Our guest speaker is Professor Mark Prausnitz. He has dedicated his career to finding innovative therapeutic solutions for global health and using various advanced drug delivery systems, and in particular microneedles, which is clearly a passion which has profoundly impacted global health. Under his leadership, the field of microneedles revolutionized the way we design 21st century therapeutics, be it for infectious diseases or oncology as well. Prof Prausnitz is a Regents Professor and entrepreneur at the Georgia Institute of Technology in the US. He established the field of microneedles in medicine and remains its leader. So not every day do we get to hear from the person who established the field. And just to know, he's known as the father of microneedles. He has published more than 340 peer-reviewed papers. These are selected obviously and basically in journals like Nature Medicine, Nature Biotechnology, Nature Nanotech, Nature Biomed Engineering, Lancet Science Translation Medicine, and others. I said Nature about maybe 5 times, so people know the caliber of the person that we hear to hear from. He has invented more than 75 US patents and is a member of the US National Academy of Engineering and also National Academy of Inventors. He co-founded 9 companies based on his research, raising almost $350 million in clinical translation work, with 1 FDA-approved product in clinical use and more in clinical development. His drug delivery systems have been studied in more than 10 phase 1, 2, and 3 clinical trials. He has founded the Center for Drug Design, Development, and Delivery, through which he awarded over 440 fellowships to doctoral students conducting pharmaceutical research. Prof Prausnitz is a passionate advocate for science and is also an inspiration to many. Today he will be sharing his insights on sampling interstitial fluid from skin as a novel source of biomarkers. So slightly different focus on drug delivery, but he will get to tell you all about that. So get ready to be inspired and challenged, and please join me in giving a warm welcome to Prof Prausnitz. Thank you.
Thank you for that overly generous introduction, and thanks everybody for coming here. So as mentioned, what I'd like to talk to you about is sampling interstitial fluid from skin as a novel source of biomarkers. But maybe before I jump into that, just a brief comment about why I'm here. So I teach at Georgia Tech, so the university in Atlanta, an engineering, science and engineering oriented university. I'm an engineer by training. And I have the opportunity for a sabbatical. So I'm off from teaching this semester, and so I'm spending 2 months here in South Africa. I'll also be going to India and Brazil as part of the sabbatical as well. So my goal while being here is to tell you about what we're doing, but I'm at least as interested to hear what you're doing. And part of my objective for today is to try to show you some of the capabilities that we've developed and try to understand how they might be useful, how we might collaborate using those to achieve scientific and medical objectives that you might have. So please do share your ideas, and hopefully some collaborations will come out of this visit. So, sampling interstitial fluid from skin. So interstitial fluid, this is the fluid that sits in the tissues of the body, and the skin among others. And we think this could be a novel source of biomarkers, other biomarkers for diagnosis, for disease monitoring, for physiological research, biomarkers that you may not find in the blood or may be inconvenient to find in the blood. So we think that this is a new kind of fluid that is very under-researched, and I'll explain why that might be. I do need to give a disclosure. As I mentioned, I started some companies and have a financial interest in some of the things we're going to talk about. So the microneedle is the enabling technology that I'll be focused on. And I wanted to give a little introduction to the microneedle first before we jump into how we're using that to access interstitial fluid. And these microneedles come in a variety of different forms. I've shown the 4 main types that are used for drug delivery. One of them is the solid microneedle. So you make some needles that are hundreds of microns long, and they are able to be pressed into the skin, poking holes in the very outer layer of skin, which is the stratum corneum, and that's the barrier layer. That's the reason why things don't just get readily absorbed into your body. And then drug can diffuse in through the holes that you have made. Another possibility is coated microneedles. You could take these microneedles, put a coating on their surface that has your drug in it, press the microneedles into the skin, the coating dissolves off, and a few minutes later you pull the patch away, and the dose has been delivered. So the first case is more of a slow, steady delivery into the skin. The second one is an immediate bolus delivery into the skin. You can take that coating, and rather than coat it on a microneedle, you can turn that coating itself into a microneedle. So now you have a microneedle that's completely made out of water-soluble materials. So when you press it into the skin, again, the drug is released as it dissolves, and there's no sharps waste when you're finished because the needle has disappeared. The final example would be a hollow microneedle. So this is more like a hypodermic needle. Now you have a hollow bored needle, you have a liquid formulation, the liquid flows through the interior, and you make an injection. And you can target that injection, for example, into the skin in a reliable way. So these are all in various stages of study and development. The solid microneedles, for example, they're actually in widespread use in cosmetics. Search under microneedling or dermaroller on Google, and you'll see many examples of these. Actually, the very first one developed was by a doctor in South Africa. But there was another group in Germany, and they were better at marketing, apparently. And so the dermaroller of Germany lives on, and I think the South African version doesn't, as far as I know. There is this very early connection to South Africa. And the hollow microneedles, they also have been an approved product for administering flu vaccine to the skin. Not a commercial success, so it's still not going, but it did receive regulatory approval and was used for a period of time. So these are some different variations on a theme. The common theme is you've got this thin barrier you need to get across the stratum corneum. So why make a big hypodermic needle? Make a very small needle to cross that barrier and get into the body in a patch-like format. So here are what some microneedles look like. In the foreground is a collection of 10 of these microneedle patches. The background is a multi-dose vial and some needles and syringes, the competition, if you will. We could then zoom in on one of these patches, and now you can see a little better what's going on. There's an array of 100 green dye-filled microneedles. There's a shiny area around the periphery. That's an adhesive, so it will stick to your skin. There's a tab that doesn't have adhesive. That's for handling purposes. And then you zoom in a little further, and you can see the actual microneedles now. You can see their dimensions, so they're hundreds of microns in dimensions. And the green dye representing the vaccine or the drug, it is focused in the needle itself. We don't want it in the backing of the patch because it's wasted there. We want it to be in the needle tip, and I'll show you momentarily how we do that. As we developed these patches, we had a number of considerations in mind. So from a patient administration standpoint, we want minimally trained personnel, which can be patients themselves, to be able to administer these patches. And as you can see in the picture, it's simply a matter of pressing the patch to the skin, leaving it, and then pulling it back off. Something that people can learn with no training. There's no applicator, it's a thumb. There's no reconstitution. That is, we have the stability of a dry formulation, but you don't have to add water to it before injecting it. You administer it as a dry formulation, and it reconstitutes within your skin. The skin's fluids wet it and dissolve it. It's a painless delivery. There's no fear of needles, so that can facilitate acceptance by patients. The manufacturing process is a low-cost and scalable one, and it basically looks like this. We start out with a mold in the inverse shape of the microneedles. We then cast the formulation that has the vaccine or the drug in it to fill the needle cavities, thereby making the needles. We then do another cast which has material in it other than the vaccine or the drug, and it dries to make the patch backing. And once the whole thing is completed its drying process, you peel it out of the mold and you're done. So this two-cast process is how we focus the drug or the vaccine into the microneedle and not in the backing. And the process as a whole, you mix up two solutions, you cast them as two films. Once it's dried, you take it out and you're done, you package it. So this is something that the expectation is in mass production can be less than $1 each to produce. So it's something that is feasible and within the economics of many healthcare systems. From a transportation and storage standpoint, there are advantages such as a small package size. You don't have needles and syringes and vials, and notably a lot of water. Most of what gets injected into people is water with just a little bit of the drug in it. So we don't need or want any water, so we can keep things very small. And there's also the improved thermostability, that is, we can get at least partially, or in some cases fully, out of refrigeration during storage, and that can make the logistics much simpler. From a waste disposal standpoint, if we're using the dissolvable microneedles, there's no biohazardous sharps waste, so you don't have to dispose of it accordingly. It's impossible to reuse because the needles are gone. There's just nothing left to reuse. And whatever disposal volume there is that remains is very small. So all of these things have informed our design of these microneedles, and again, especially in the context of drug delivery. Our most advanced work in this area is with vaccines. We've completed a phase 1 clinical trial on flu vaccination in 100 subjects. That was done in Atlanta. And more recently, we carried out a phase 1/2 clinical trial on measles and rubella vaccination. That was done in the Gambia in an age de-escalation manner. So we started in adults, then went to toddlers, and finally went to infants. And in both of those studies, it was very well tolerated, and the immune response was similar, or in one case better, than the comparator, that is the injection of the vaccine conventionally. So that's just a little background. I'd like to now switch gears and not talk about using the microneedles to deliver drugs into the body, but I want to flip it around 180 degrees, and I want to use them to pull things out. So why might we be interested in this? So if you think about the various biomarkers that are collected in medicine, most of them come from blood. There are also some secreted fluids, urine or saliva and so forth. A small number of things are measured there, but blood is, of course, the workhorse. Blood is only about 8% of the fluid in the body. There's lots of other fluid there. So what about those other fluids? Is there something interesting in them? Well, about half of that fluid is intracellular fluid. That's very hard to access without killing the cell. But close to a quarter of that fluid is the interstitial fluid, or the extracellular fluid. This is the fluid that bathes the cells of our tissues. It has information related to blood, because that's the source of that fluid. It has information related to the cells that it's bathing, because those cells are metabolizing. So there should be some information that's similar to blood, but there should also be information that is distinctly different from the blood. So we thought about how could we access this interstitial fluid. Of course, if you pierce your skin with a needle, if you pierce it hard enough, it bleeds. If you do it very gently, very superficially, nothing comes out. How do you get this fluid to come out? It doesn't just readily flow. So that's going to be a fair amount of what I'm going to talk about, is how we can get the interstitial fluid out, and that then presents some opportunities to do things with that fluid. So just a little side note on the interstitial fluid and what it is and where it comes from. So the capillary and the oxygen nutrients to the cells of the body, they leak out, carrying all those things with it. And that which leaks out of the capillaries is this interstitial fluid. Then flows through the tissue, headed to the lymphatic system, where it then gets taken up. And en route, it encounters cells, and it delivers oxygen, and it takes CO2, and it delivers nutrients, and it of the interstitial fluid as it passes through the tissue environment. So this is the very fluid that we seek to access. So a few more thoughts on why we care. Why are we spending time on this? So, and then we have a few bases for evaluation. So one is, are there systemic biomarkers, things that are currently measured in serology? Well, obviously blood does very well with that. And I gave interstitial fluid 2 stars because, as you'll see, it has a lot of the same information that you can find in the blood, but doesn't have all of the same information. Does it contain tissue biomarkers? Well, blood is not so good at that. I mean, obviously, if there's a biomarker in the tissue, it probably will flow out of the tissue and end up in the bloodstream, but in a dilute form, and so it may be harder to access. Whereas the interstitial fluid, that's collected from the tissue. That's exactly where you want to get that kind of a biomarker. What about clotting? And clotting is particularly interesting if you want to do a continuous or repeated measurement over time. Well, blood, of course, clots, and having some indwelling mechanism of accessing blood is, I mean, you can have an IV in the clinical environment, but at home, that's really not going to work. In terms of interstitial fluid, it doesn't clot the way blood clots. So continuous sampling is a very reasonable possibility. Does it contain cells? Well, sometimes you want cells because that's where your biomarker is, but assuming you don't, then you probably don't want the cells there for analysis because they're going to oftentimes mess up the analytical tools that you were using. And so interstitial fluid, for better or worse, if you didn't want the cells, for better, it doesn't contain cells. It's just a fluid. And finally, easy to access. Well, blood's pretty easy to access. I mean, it's if you need a venous puncture, I mean, that's a little harder certainly to do at home. You could do a finger stick at home. So I gave it two pluses. Interstitial fluid, at least today, there are no ways to access it, and that's what we seek to change. So why is it so hard to collect interstitial fluid? Well, there are some methods that are used. One method that's used in a research context is suction blisters. In this method, you put a device on the skin that applies heat and applies negative pressure, applies suction. You leave it there for 45 minutes or an hour. It literally makes a blister, just if you're out working too hard and rubbing the skin, somehow you get blisters. This sucks up a blister full of fluid. You then pop the blister, you pull the fluid out, and that is something at least related to interstitial fluid. Obviously, you've done some damage to the tissue in the process. It leaves an unpleasant appearance on your skin for a number of weeks as it heals. It's a limited research tool. There's also microdialysis and some related techniques where now you put plumbing in the skin. And so through a minor surgical procedure, you put microdialysis tubing into the skin and you run fluid through and you then collect biomarkers that are in the skin in that fluid and take it out. And again, in a research environment with trained technicians who know how to do this, this is a method that can be done. But you're not going to do it at home tonight. Reverse iontophoresis is something that has been developed, and this is where you use electrophoresis. So as you know, in electrophoresis, you have a charged molecule, an electric field. It moves in that electric field. So you can use that same technique to move molecules out of the skin under the influence of an electric field. And in fact, there was a product called the Gluco Watch that would pull glucose out of the skin for diabetes monitoring. It was ultimately not a commercial success, but the technology was developed. And it's very limited what you could do with that kind of an approach. So people have tried, but it's just not easy to get this fluid out. So our approach, of course, as you know, we're going to be using the microneedles to access the interstitial fluid. We're going to puncture the skin with the microneedles in a way that makes channels for fluid flow, channels of micron dimensions. So as far as any molecule is concerned, there's no sieving effect. Molecules are nanometers in dimensions. We've got holes of microns dimensions. So molecules can fit through. There's no problem with that. But because these microneedles are small, the odds of them hitting a blood vessel is reduced. So they're narrow and they're shallow. And so by doing things in the right way, we think we can make holes in the skin that access the fluid, not cause bleeding in the process, and then get that fluid to come out. So that's the value proposition that we offer. So an outline of what remains is 3 different parts on this topic. First is the mechanisms to drive interstitial fluid into the microneedle. So I sort of gave you the high-level idea of the microneedle, but how do you actually get that fluid to flow out? Second is then collection and analysis of this interstitial fluid using the suction-based approach, and I'll show you some results from a human study that we did. And then finally, another method that we've developed is collection and analysis of ISF without using suction, just using a microneedle device by itself. So let's think about how we might get this fluid to come out of the skin. One way could be to use a hydrogel material. So you have a gel that is cross-linked, but it's dry. And you stick that into the skin, it contacts water, and the water then gets sucked up into the hydrogel swells, and then you can pull the microneedle patch out, and you have a swollen microneedle, swollen with interstitial fluid. And you can perhaps see in the picture the microneedles before, and then you can perhaps tell that they've swollen some in the process after being put into the skin. Another approach we might use is capillary action. That's where fluid will be sucked up into a small channel by the surface tension forces, And so we could take advantage of that by making channels, and then the way we made the channel is either, as shown on the bottom, a hollow microneedle that will suck the fluid up, or we made it with 2 microneedles and a filter paper between them, and the porosity of the filter paper sucks the fluid up. A third approach we could use is osmosis. So osmosis is where you have a high solute concentration on one side and a low solute concentration on the other, and either the salt, let's say it's a salt, the solute, in any case, the solute can diffuse from high concentration to low concentration, but also the liquid, say the water, can flow from the low concentration of solute to the high concentration. So both of those things will take place. So we can take advantage of that, a microneedle that has lots of solute in it, and it'll be transient. The solute will diffuse away, but before it's all diffused away, water can get sucked in the process. And then finally, we can apply a pressure. Either a positive pressure, squeeze the skin, or a negative pressure, pull a suction on the skin. Not so much that you get a blister, but a suction to pull the fluid out. So we investigated these 4 different approaches to mobilizing the fluid in the skin. In terms of the hydrogel, if we think about in all of these, there are 3 steps that could be limitations in flow of the fluid. Step 1 is flow within the dermis. So the fluid has to flow within the dermis of the skin and make its way to the microneedle. The second step is it has to partition out of the skin into the microneedle. And then the third one, it has to flow up into the microneedle and be collected. In the case of a hydrogel, the process is diffusion. And so you're not enhancing any of those steps. You're just providing a place for water to diffuse in and carry biomarkers with it. So that we expect actually to be the slowest. If we move to capillary action, now there is a force that is sucking fluid up through the microneedle, that surface tension capillary force. But we're not doing anything to the skin, so the capillary action should be better than diffusion. Osmosis is a similar thing. The osmosis is really going to act just locally near the microneedle because the concentration gradients will drop off quickly. But finally, when we get to the use of pressure, then that pressure is not just in the needle, but we're applying a pressure to the skin around it. I still have a red color on step number 1. It's not as dark as it was before. So the application of a pressure is going to help move the fluid through the skin as well as up through the microneedle. So with that conceptual information in place, we made some measurements where we took microneedles that we made of each of these designs, we pressed them into, this is in pig skin ex vivo, and then we measured how much fluid that we got out. And here what is shown is, note it's on a log scale on that x-axis. At the bottom is the hydrogels. Then when we make either micropores, porous microneedles, hollow microneedles, those are all different ways of getting capillary action. We're getting significantly more. We then go to osmosis, which is kind of comparable or more. And then finally, when we apply either positive pressure or negative vacuum pressure, then we get another increment better still. So the quantitative data agree qualitatively with those explanations that I gave before. So you can imagine the conclusion we draw from this. Pressure is good. We want to give pressure. We want to mobilize the fluid in the tissue as well as in the microneedle. So that led us to the next part of the presentation, which is collection and analysis of ISF by suction. So we're going to use that negative pressure to mobilize the fluid. And the process looks like this. We'd have microneedles, we stick them in the skin, we pull them out, so they're just pokers, just poking some holes in the skin, and then we put on a cup on the skin, apply a vacuum, and coax that fluid to come out. This is what the microneedles look like. So these are made out of metal. They are made of a stainless steel sheet that we cut with a laser or etch them by a chemical method. They're short, so they're only 200 microns long. So we want to get into the skin, but we don't want to go too deep, because the deeper you go, the more you're going to hit blood vessels. There are no blood vessels in the epidermis of the skin, the upper 100-ish microns. The blood vessels are about 150 microns deep. So these needles are just getting close to that capillary bed, but trying to minimize puncturing the capillaries because we don't want blood. We use this vacuum device. It's actually the device that is sold to make suction blisters, but we turn it down a little bit and we don't make the suction blisters. We also don't use the heat. And then here you can see we put some tegaderm, sort of a plastic film on the skin. We've cut out 3 circles, exposing the skin there, and then you can see some spots of interstitial fluid. They're a little hard to see because it's clear. They're not bloody. It's clear fluid, and there are these droplets that have accumulated on the surface of the skin. So let me show now some results of using that technique. And this is actually from our very initial studies. I believe this is the first paper that was published on accessing interstitial fluid out of the skin using a microneedle technique back in 2005. At that time, we used a microneedle that was made out of glass. It was a micropipette. Maybe some of you use micropipettes in the lab for intracellular recordings, or it's used in vitro fertilization. So we made a microneedle by that method. That was a convenient way. We then press into the skin, and you can see we've imaged one of the pores, one of those puncture holes in the skin, either viewed above or histologically from a side view. These needles are going in deeper than the 200 microns that I mentioned before. And now, again, you can see some fluid that we have pulled out. Using that approach, we looked at glucose concentration. And this is what's presented here is something called a Clark error grid. It's commonly used when people want to analyze whether a glucometer is working correctly or not. The y-axis is the glucose level as determined in our interstitial fluid sample, and the x-axis is the blood glucose level as determined by a conventional glucometer after finger stick. And so if they agree with each other, there's this 45-degree line down the center in the A region, and that would be perfect agreement between the two. The scatter of points can get into the B, C, D, and E regions. Those regions, from a clinical perspective, get progressively more dangerous for the patient. So as you can see, most of those points are in that A region that glucometers are supposed to be in, 96% of the points. These are all done in rats. There's also on the second graph are just a few data points. I think it's 6 data points. That was done on humans. These were all normal human subjects, so they were not diabetic. So there you see their blood glucose level in all cases is right about 100, as it should be. But in any case, the conclusion was we can get interstitial fluid from humans, from rats. We can measure glucose in it, and we get a number that's generally within the range that the FDA would expect you to have with a new glucometer. We also looked at a time course, so we monitored glucose concentration in the rats. Turns out when you give rats the anesthetic that we use, the anesthetic causes blood glucose level to go up. And so we monitored them, we gave the anesthetic, their blood glucose came up, we then dialed down the anesthetic and the glucose level came down, so it was a convenient artifact of the anesthesia, and so we were able to monitor the kinetics of the glucose concentration, and when we do it, it's not labeled, but one of those curves is the blood and the other one is the interstitial fluid. You can see they track each other very nicely. So that led us to more recently do work on human subjects where we collected 3 different samples. We collected either the interstitial fluid with these smaller metal microneedles that I showed you before, or we collected blood plasma by venipuncture. We also collected the suction blister fluid by the method that I mentioned. This is in 21 healthy adults. And what we can see when we make this Venn diagram of all of the biomarkers that were detected, and this is more than 10,000 different compounds that were detected by mass spectrometry analysis, you can see that 69% of the biomarkers that of the compounds, I shouldn't necessarily call them biomarkers, but of the compounds that we identified, they were common to the plasma and the interstitial fluid. So that tells us that if you're looking for some biomarker that's in the blood, there's a good chance, not a perfect chance, but there's a good chance that you will also find it in the interstitial fluid. I think a reasonable expectation is that the molecule is not too high molecular weight, so maybe 100,000 kind of molecular weight or less. That's going to cross across the capillary wall and enter into the interstitial fluid out of the blood. And if the molecule is hydrophilic or otherwise is not bound, if it's bound to cells or it's bound to serum proteins, that's going to keep it in the blood and make it harder to get out. So I don't have the data to support this, but my expectation is that the 11% of compounds that were in the plasma, not the interstitial fluid, were either too high molecular weight or, in this case, were probably bound to something in the blood which inhibited their leaving. But interestingly, there's another 17% of these compounds that were in the interstitial fluid and were not in the blood. So that's telling us there could be some new biomarkers that we don't know about. We haven't been able to see them before because they don't appear in the blood, that we are able to find now in the interstitial fluid. So let's go a little further with this. If we look at some of the biomarkers, so we detected this 10,000 plus compounds as peaks out of the mass spec analysis. We don't know what all of those peaks are. There are some unique molecules, but some subset of them have been identified, and we know exactly what molecule it is. And here are some of the different classes. So you can see amino acids and their metabolites. There was more than 50 of those biomarkers were in that category, and you can look down the list yourself and see the various other ones that we had. We then looked to see in which cases there was a high prevalence of that biomarker in the plasma and the interstitial fluid, also the suction blister fluid, which isn't as interesting for today. And as you can see in these green cases, there were ones where we were able to find them, 100% of them were in the interstitial fluid, but only a smaller fraction of them were present in the plasma. And then likewise, there's a case here where it was the reverse, that we were more successful to find the sterile metabolism molecules in the plasma than we were in the interstitial fluid. A few other things to note about the biomarkers that were unique to the interstitial fluid. So a number of them were related to nucleotide-related metabolism. So that's indicative of interaction with cells, the metabolic processes happening in the cells. There were some dermatologically relevant molecules, namely structural components of the skin, so indicating the potential to monitor local biomarkers. And there were some exogenous molecules like pesticides and phytochemicals that we saw as well. So this is only the beginning. We have some snapshot into what's going on in the interstitial fluid, how it relates and how it doesn't relate to what's found in the blood. So we went on now to have a look at what happens in humans over time. So I showed you the glucose result. We then added on to that delivery of Atlanta's favorite drug. So you may know Atlanta is the home of Coca-Cola, and so caffeine is our drug of choice. And we gave subjects caffeine to drink. We gave them Cokes. We either gave them regular Coke, I should say diet regular Coke with caffeine, or we gave them caffeine-free Coke as our placebo negative control. And then we measured the caffeine level in interstitial fluid and in the plasma. And you can see the dark colored data points. Those are both the caffeinated beverage group, where measurements made in interstitial fluid and plasma fall on top of each other. And there's, of course, the background from the caffeine-free Coke with the white data points. We determined the various pharmacokinetic parameters, and you can look at them if you want, but the main point is, if you do a statistical test, the PK parameters in the plasma and the interstitial fluid are statistically indistinguishable from each other. So this suggests for a small molecule that is water-soluble, like caffeine, readily crosses the capillary wall, we are finding it in the bloodstream and in the interstitial fluid equally well. That brings me to the last topic, which is, maybe you don't want to apply suction. It's a little cumbersome. You stick needles in, you take them out, you put on a suction device, you take it off, you collect the interstitial fluid. Great research tool. People aren't going to do exactly that at home, although you might be able to build an all-inclusive device that does those functions transparently, so the patient just pushes a button and the rest happens inside the box. But we thought maybe we can get rid of the suction and make it easier. And the design looks like this, where we have an array of these microneedles. So this is 9 of them all in a row. These are the metal ones again. And we have a strip of filter. I'm Am I still on? Should I keep talking? Yeah, keep going? All right. Wake up. A strip of filter paper on the back that we calibrated the size of it so that when it becomes completely wet, it holds 1 microliter of fluid. And so what you can see is, so A shows that, B shows a close-up of one of these microneedles, and then C shows us applying the microneedles to the skin of a hairless rat. And so the needle's being pressed in, and we're actually pumping it back and forth a few times, like 5 or 10 times. And you can see the shiny part on there where the fluid has come up. So you keep doing that until all the fluid is wet, and then you stop, and then you know you've collected your microliter. We also made a bigger piece of paper, and that was a 2 microliter. So a very simple method to collect the fluid. We found, perhaps not surprisingly, that the number of times we stick the needles into the skin will give us more fluid, from 1 up to 20 times of inserting into the skin. We also varied some things about the microneedle, the number of needles and the thickness of the metal, which had a lesser effect than the number of times that we inserted the needles. So then we made an unexpected discovery, and bear with me as I lead you through the data. So in group number 1, we took the microneedles and stuck them into the skin back and forth 10 times, kind of pumped the fluid out onto the filter paper. And we found by doing that, there was a little more than 1 microliter of fluid that we were able to collect. We then did a next experiment where we took blunt microneedles, pressed them against the skin 10 times. They didn't penetrate, they just bumped against the skin 10 times. We waited 5 minutes, and then we did our 10 microneedle insertions again. And as you can see, that made no difference. Those blunt microneedles pressing against the skin were not relevant. Then we did another experiment. We did our 10 microneedle insertions. We collected some fluid, a little more than a microliter. We waited 5 minutes, and then we went back to the same place, and we went and collected again 10 times, and we got 5 times more fluid to come out. So we had anecdotally observed this when we were just doing our experiments. We'd collect some fluid. We'd come back later and say, hey, let's collect some more, and do it at the same site. We got much more fluid. What's going on here? And so what we think is going on is edema. That the first collection that penetrated the skin, it caused some minor tissue injury. And so there was some more fluid flowed into that skin, causing an edema, causing some swelling. So when you come back the second time, there's more fluid in there, and the fluid more readily flows, and you can get a lot more to come out. So to test that hypothesis, we did these experiments, and then we excised the tissue and measured the water content of the tissue. And as you can see, group 3 has a lot more water in it than group 1. For reasons we don't completely understand, group 2 had a little more water in it also, but in any case, we think this is consistent with the general idea that increasing the hydration in the tissue can increase the amount of fluid you get out. And in fact, there's a follow-on student who has made this, the focus of his PhD is to study the skin hydration and to see how that influences the fluid that comes out, and the hypothesis continues to hold up. We looked at the tissue afterwards to see if we saw any evidence of damage, and basically there's nothing notable to see in these histological sections. The skin seems to tolerate it very well. Also by clinical exam, there's just this mild edema that then clears quickly afterwards. So we use this kind of an approach now to sample, and in this case we looked at vancomycin. Vancomycin as a drug, so a real drug, not caffeine in this case, and we administered vancomycin to rats and then monitored either in the case of the white squares, that was the serum concentration, or the black data points is the interstitial fluid. And this time you see something a little different. Unlike in the caffeine study where everything tracked very nicely, if you look at what's in the serum, there's a sudden increase in the vancomycin concentration, which then decays over time. This was an IV injection. And so that's what you would expect after an IV injection. You dump all the drug right in the bloodstream, and then it gets cleared out of the body over time. In the interstitial fluid, it's more blunted. You don't see that peak followed by a valley, but it's more of a plateau. And what we think that reflects is that the drug is immediately in the bloodstream, and it takes some time for it to leak out of the bloodstream and get into the tissue and accumulate in there, and then you can measure it. And so we think that this difference between interstitial fluid and serum, it's not a bad thing, showing that the interstitial fluid can't mimic the serum. It's actually showing us the physiology of what goes on when you give a drug IV. We did another experiment where we vaccinated rats against polio, and then we came back later and measured antibodies present either in the serum or in the interstitial fluid. And you can see at 0 and 1 week, there's no antibody response yet. And then at 3 and 4 weeks, the antibody response kicks in. The antibody level in the interstitial fluid is a little lower. It's about 3/4 as much as what we found in the serum. And that is consistent with some other limited analysis that exists on what's found in the interstitial fluid. As molecules get very big, sometimes as antibodies are, then the concentration in the interstitial fluid can be lower than what's seen in the blood. Let me play a video, and so this now comes from the work of the graduate student I mentioned, who has gone further in terms of getting a good edema response in the skin in a controlled way. And the video, it's a 1.5-minute video, but I sped it up tenfold. So what we're looking at, actually I'll tell you what this is imaging. So this is the skin of a human subject, and we're using a device that we bought on Amazon for $25. It is a blackhead remover. So this is a device that you put on the skin, it applies suction and pulls the blackhead out of your skin. But what's so great about it is that because everybody wants to see the blackhead come out of the skin, it has a built-in video camera. So you can post your blackhead removal on Instagram. So we didn't use it for quite that purpose, but hey, this was great. We had a suction device that has a camera in it. So you are going to watch the skin surface after we have pre-treated the skin with the microneedles, and you'll see the fluid come out. It's a little bit blurry because the camera isn't focused in exactly the right place, but let's get it started. Hopefully you can see these little droplets forming and lots of fluid being sucked out of the skin. And so all that happened within a minute and a half. So you could have a very rapid collection. This particular time, it was about 15 microliters of fluid that we pulled out. We've gotten as much as more than 100 microliters from some subjects. We don't know why some subjects give us more and some subjects give us less. We don't know the degree to which it's something that we can control, the way that we're using the microneedles, the suction, whatnot, and the degree to which it's just, some people are leakier than others. So that's work that continues. So that brings me to the end. Hopefully I've given you some sense for these microneedles, what they might be possible for. One of the things they might be possible for is collecting this interstitial fluid from the skin. We've explored various mechanisms. We found suction to be particularly useful, but also we found edema in the skin can be helpful as well. And in terms of the next steps, what we've been really focused on is developing a device and method for simple interstitial fluid collection. But where we're going is we want to use this then to enable researchers, perhaps some of you, to study interstitial fluid and identify its greatest value. Until now, there hasn't been a way to get the fluid, so people haven't studied it. All right, we've got the fluid. What do we do with it? So we're trying to learn that through collaboration. And then hopefully some things will come of it. There'll be some things interstitial fluid is good for. There'll be some things it probably isn't good for, but hopefully there are some things that it is good for that may then be useful as a source of biomarkers in clinical medicine. And it may even be that people can collect this interstitial fluid at home with a simple patch, and this could enable not only diagnostics of special biomarkers, but a more routine and democratized access to these kinds of diagnostic and monitoring capabilities that today might require access to a healthcare facility. So with that, let me acknowledge the research team that has worked on this, the graduate students and postdocs in the group, and the funding sources that have enabled it as well, and see if you have any questions or ideas. So thanks so much.
My question is, after you have put the micro needles onto the skin, right, and then you put the suction thing to pull out the fluid, is there any point where it interacts with sweat or anything like that, or do you clean up the area before you actually put in the microneedles? And also, since it's metals, do you feel pain or? Yeah,
Yep.
that's my question.
So the procedure involves first that we will clean off the skin with an alcohol wipe. Then we'll press the microneedles in and pull them out. People can feel the microneedles. They feel a pressing sensation. In other studies that we have done where we've taken a patch that has no microneedles on it and just pressed the flat thing against the skin versus microneedles that actually puncture, people can tell the difference. They feel something more is going on there. They generally don't describe it as painful or unpleasant. It's just they feel something is happening. So then we press the needles in, pull them out, apply the suction, and then at that point the fluid will collect up on the skin surface, and we'll take a gauze and collect all the fluid on the gauze for analysis.
I just have a question about the sampling. Is it, from what you're saying, as I understand it, are we limited by the size of the biomarkers, that it can't actually come out of the capillaries through the extracellular matrix into the interstitial fluid? And then also, with the dermis, there are lots of immune cells present. So is there an inflammatory response by using the microneedles? And if it is, then are there inflammatory biomarkers that you can detect? And then will that not be a false positive as to what's actually there?
Yeah.
Thanks.
In terms of the size of the molecule, so if the molecule is not able to leave the capillary and enter the tissue, we're of course not going to detect it. So that's a physiological issue that we can't control. So that's something you have to understand and design your system accordingly. If the molecule is able to escape the capillary, or if the molecule is produced in the skin itself, so it could be very big, but produced by cells in the skin, then the limitation is, can it get to the microneedle? So now it has to make its way through the extracellular matrix, and so if it can make its way through that, then once it gets to the microneedle, I think we don't have size limitations. The hole that we make is microns. It's going to be tens, if not 100 microns in width, and so things will flow through that hole quite readily. Your second question was about inflammation. So we've pressed microneedles a lot into the skin, more in the context of the drug delivery side of things, but anyway, the injury of a microneedle going in the skin is the same whether you're putting stuff in or taking stuff out. And what we see very reliably is you press the microneedles in, you pull them out, and immediately after you don't see anything. You actually kind of see a little indentation of the skin because you've just been pushing on it. And then after a couple of minutes, then very mild erythema sets in, which is punctate. So at the site of each microneedle, and so typical spacing might be 1 millimeter between each needle. So you see this very faint pink dot. You see 100 of them in an array where each of those microneedles went in. It never merges together into a broad erythema. It's always just a highly localized spot. And again, a faint pink. You have to look carefully to see it. And then within a few hours, there's nothing to see at all. It goes away. If we are trying to cause edema and are a little more aggressive, so then we do cause edema to occur, and it's visible and palpable. You can tell there's a little bump of fluid there, but that will resolve in under an hour. Oh, I'm sorry. There's a little more to your question for the inflammatory biomarker. So if your interest is to detect some cytokine, then you do need to be careful, because you may upregulate some cytokines due to this micro injury, and so you have to account for that.
Yes, please. Yeah, mine is just comments. I wanted to comment on the slide that you put. You were looking at antibodies, and then you looked at, I think, week 1 and week 2 and week 3. I just wanted to comment that in vertebrates, in humans and in mice, it takes around 7 days for the body to make immune response. So probably that's why you couldn't pick it on the first week. And then the second comment is that when you prick the skin to get the CSF, right? Interstitial fluid.
ISF, yeah.
So the matrix that is going to be on the first prick will not be the same as the matrix that will be in after some time, after 5 minutes that you indicated. So researchers need to be aware of the fact that, as you just mentioned now, there will be a cytokine cascade, and therefore that will interfere with the matrix. And then there is 2 more, hope I didn't forget. Yeah, I had 2 more comments, slipped my mind. It will come back.
We have a different section after this as well.
All right.
Yeah, so I think if you're going the Heisenberg principle that if you perturb an environment to make by making a measurement, you perturb the environment, and by definition you've altered it. So it's true. So we try to minimize that perturbation with the needles going in, but yes, we perturb the environment and it could have an effect.
Yeah, I just want to know about other surfaces. So obviously mucosal surfaces are far more interesting to some people than others, in particular around looking at immune responses post-vaccination. So have you thought about using other mucosal surfaces? Obviously the easiest one would be the mouth, but for other people who are working in STI vaccination, other mucosal surfaces like the cervical vaginal area, and could there be an application in other surfaces, because this seems like less traumatic than a biopsy, and one could think about using that for looking for immune responses post-vaccination.
Yeah, I think that is possible. There has been work done with microneedles pressed into wet mucosal surfaces in the context of vaccination, say in the mouth. So yes, microneedles can penetrate those. Our standard method involves bringing the interstitial fluid up to the skin's surface. And if that surface is covered in moisture, that might be a problem because it's going to get diluted into and mixed with the saliva, say, that's in the mouth. So we might want to use a different microneedle. We might want to use, for example, say the hydrogel microneedle, which collects the fluid within the skin, and then you pull it out, and it doesn't risk contamination of whatever's on the skin or on the mucosal surface. So we need a little bit different thinking, but I think it's definitely feasible.
Thank you. We have a question from one of our online attendees. So Stian Dabaskakni is asking, can it be used to diagnose skin cancer, or has it been used for this purpose before?
I think that's an excellent application. It's something that we are looking into. For diagnosing skin cancer, sometimes it can be done by a good clinical exam, but in other cases biopsy is needed. So we think that this could be an alternative to biopsy. Would be, I think, preferable with essentially all patients not to have a biopsy if they could avoid that. But there need to be the molecular biomarkers established that will provide the reliability of a diagnosis, say, for melanoma, which is currently done by biopsy.
Thank you. Mark, that was wonderful work and wonderful presentation. This issue of perturbation-induced inflammatory markers versus what would naturally be occurring during an infection could be studied. I mean, we could set up such a study with controls and people have various types of infections. One of the reasons I think this could be important is that in many parts of Africa, when a patient presents with, for instance, community-acquired pneumonia or sepsis, there's a lack of information about that would help the community health worker or the clinical officer or the physician to determine whether or not this needs to be treated with an antibacterial or not, or something else, or is this going to lead to something serious? So you can imagine your system being used in a setting like that, because presumably it is, stable. You would have those in a box somewhere. You apply, you collect the fluid, but then you'd have to have the ability to measure the cytokines. But I wonder if this issue that you brought up could be studied and determined the difference between what the patch itself does versus, what happens during an infection.
Yeah, I agree. We haven't done anything in that direction, but I think it's an interesting one.
Hi, thank you very much. That was a wonderful presentation. So just thinking about the implications for vaccine delivery, I mean, have you given thought as to how this microneedle strategy could change the way we dose vaccines? Right, so right now we give a single bolus and you'd boost, a year later or 2 years later. But with this type of technology, you could almost micro boost. So the vaccine gets released over a week as opposed to a single, and the way the immune system sees the vaccine now is completely different. Or you have one bolus and then there's something that coats the needle and protects it, and then 2 weeks later you have another bolus. Have you given thought to this?
Yes, so there has been an interest in having having vaccines administered in novel ways. So, I mean, our standard, as you point out, is you give a dose, and then a month or whatever later, you give the next dose. And there are many possible delivery scenarios. So we did a study where we didn't formulate the microneedles to have a slow release, because that's a lot of work. So we wanted to know what the release should look like. And so we applied patches onto animals either every day or every other day, went anywhere from just a bolus at the beginning to extended over a month of delivery profile. And we looked at different delivery profiles where we either gave all the dose at once, or we gave the dose, half the dose at the beginning, half the dose at the end, so prime boost, or we gave a steady dose over time, or we gave an increasing dose over time, or a decreasing dose over time. And we found that if you gave a steady dose over time or an increasing dose over time, a single dose spread out over time was as good as a prime and a boost. But if it was a decreasing dose over time, it was no better than just the prime. So the delivery profile matters. And we are working on microneedles that can have a controlled release, where the steady release is actually in the context of a long-acting contraceptive that is slowly delivered for 6 months. For vaccines, it's a lot harder to have a steady release, and especially to have one that might increase over time. But what we can have is a self-boosting scenario, where the microneedles have half of the vaccine in readily soluble form for the prime. The rest is encapsulated inside a biodegradable microneedle that breaks off and stays in the skin, and it biodegrades slowly with nothing coming out until it biodegrades enough that the interior, the core inside of the shell, becomes exposed, and then your boost comes out, say, a month later. So that's something we're working on. We're not quite there yet, but we're working on the technology to have this kind of a core shell structure that will sequester the vaccine until a later time.
Thanks, Malik. I think there is time for maybe 15 more questions. That's why we have the networking session after this. Let's just, Kate, can we go 1, 2 burning? Yeah, let's keep the conversation going and throw some ideas. Yeah.
Stay if you can, but if you have places to go, please do. Yeah, I'm happy to stay here.
Firstly, thank you for your presentation. I'm interested in the biomarker side that you've been detecting. So it's quite a multifaceted question, actually. The first is, what is the stability of the biomarker that you are detecting? Because there's multiple steps for it to penetrate, into the skin, to the microneedle first, and then through the skin. What is the stability of those biomarkers as compared to the serum biomarkers? So that's the first part. The second part is, you're inducing edema. Is it not likely that it could be diluting the biomarkers, and that's why you may not be able to detect the same? Is it not possible that these biomarkers are being degraded, maybe by an enzymatic reaction? How do you ensure that you are inserting the microneedle into the same place, after your brief period? And is it not possible that you are looking more at the trauma biomarkers and not at what the normal states biomarkers are?
Okay, a lot of questions. Remind me where you started.
Stability of your biomarkers.
The stability. So I think that's going to really depend on the biomarker. So our usual method of collection does involve drying and then reconstituting later for analysis. And then you can store it frozen or whatever you need. So I mean, we dried them, we collect them right away, put them in the freezer, and we did all the things that we could to preserve stability. But I think it's going to really vary with each biomarker. Some will be super stable, and others will need better attention. I don't know that I have any reason to believe that a biomarker is more or less stable in serum or plasma as opposed to interstitial fluid, but there's no information about that. Then in terms of the edema and effects that it could have. So the edema is coming from the vasculature. So if the source, the molecule you're interested in has its source in the vasculature, maybe you're not diluting it. If its source is in the skin, maybe you are diluting it. So yes, something you have to pay attention to. And then, I mean, edema there's more to edema than just fluid, and then could that have damaging effects to biomarkers? Yes, we don't know. I think that in the ideal world, if your goal is to correlate with what's in the blood, whatever you pull out of the skin is identical to what's in the blood. And in some cases, like with caffeine and glucose, that was pretty much the case. In other cases, it won't be identical, either because of the physiology or because you've altered the physiology by edema. As long as it's reproducible and controlled, it's probably okay. You can then account for that difference. But where it's variable and not controlled, that's when it's a problem.
I think coming close to another burning question.
One question. So another question from one of our online attendees, Minya Gerber. Would it be useful to measure the transepidermal water loss before starting the microneedle procedure to determine the moisture of the skin to see if that has an influence in obtaining more moisture from certain individuals than from others?
So one of our hypotheses is that the reason why some people are leakier than others is that they have a higher dermal water content just based on their physiology. It's hard to measure dermal water content. So transepidermal water loss is literally measuring the rate at which water is leaving the interior of the skin and coming out into the air. And not due to sweat, but just due to the water moving across the skin. It's really primarily a measure of the barrier properties of the skin. Because the interior, it's very wet. And maybe in one person it's a little very wet and the other one it's very, very wet. But I mean, it's mostly water that's in there. And so really what transepidermal water loss is measuring is whether you've poked holes successfully in the stratum corneum. So I think that would tell us if we applied our microneedles correctly and made holes. I'm pretty sure we are. I don't think we'll see a lot of variability in that measurement. We've actually struggled some because we would like to have an easy way to measure dermal water content, which we can do if we excise the tissue, but in humans that's not such a simple matter. So if anyone has some thoughts about that, we'd appreciate it, because I think that probably is an important parameter.
Thank you, colleagues. I think
not really much of a question, much of a concern that I'm having. I'm more thinking of the limitation that it comes when we go into diagnostics for biomarkers. What happened is she mentioned something about cytokine changes, especially when you inject with the micro injections. So what might happen is we might experience a micro-inflammation, and let's say we're looking for biomarkers responsible for such as diabetes. Most of the time when we're doing that is we tend to look at biomarkers such as that are like betokines most of the times. And then you'll find that at times when we do look at that, when you do punch in, even a slight bit of inflammation does induce an inflammatory response. And some of the cytokines that we do and betokines that we do note in diagnostics of such disease diseases, we do find them in such a tiny microinflammation. So that would be something that, one thing that is I'm trying to raise is that there would be prior studies that you'll have to first do to first check if like your cytokine changes are occurring with your blanks most of the time, to try and highlight that your cytokine changes are at this great level, and then at that point on, that's when you can be able to then optimize your concentration changes and highlight how your biomarkers can then. So I think that's how you will go around such things.
Yeah, no, I agree. I think most biomarkers are not cytokines, are not associated with inflammation, and so they may not be affected. But if it is something that a biomarker that you care about is related to an inflammatory process, then you need to be very careful and do things exactly as you've described. I agree.
Thanks, colleagues. I think we're gonna end it there. Speaking the Olympics, I think doping in sport as well could be one application, and also the illicit drugs as well, which is a big problem locally as well. Using some at pharmacy level, so it's not going for the big injection and getting the blood test result later. So, Prof, maybe any comment on that? I don't want to put you now. I know you've spoken enough already, but that will end as the last question.
So I think that monitoring for drugs, whether they are like here vancomycin, or whether they are drugs that you're not supposed to have, this would be a nice way to do it. One concept is that you can put on one of these hydrogel patches, stick it on the skin, and leave it there, and it can be collecting the fluid continuously, slowly but continuously, say for a week. So if you don't want somebody to take those drugs that they're not supposed to be taking, and stop and wait a couple of days and it washes out of the system, you're monitoring continuously. Or likewise, someone who's supposed to be taking a drug and doesn't, and takes it the day before their doctor's appointment, you get information over time by collecting. So that's, I think, a possibility.
Valuable to rehabs as well. Okay, colleagues, we're going to end the session now. There's a networking session after this year. We've eaten literally and figuratively into some of it. There's food up there, and we can obviously, if you have any other questions for Prof, he's here, and he'll be around for another
3 more weeks.
3 more weeks. We're hosting him here at the university. So if pop an email if there's anything. Thank you.
See you upstairs. Thank you.